Today we will be talking about expression of recombinant proteins, specifically DHFR.
The goal of this lab is to induce the expression of the DHFR protein
and grow it in order to comprehend its biological role in a laboratory biomedical environment.
The reason why to produce proteins recombinantly is that proteins need to be purified in large quantities. However, some proteins are not naturally produced in a form and in amounts that allow easy purification for this to happen.
Hey guys, it's your favorite girl Gamora. Just got finished strangling my sister Nebula. Today we're going to talk about DHFR. So DHFR is dihydrofolate reductase. It is an enzyme that converts dihydrofolate, a folic acid derivative, into tetrahydrofolate by the addition of a hydride from NADPH.
E. coli will be used to express the DHFR gene because it amplifies quickly and is inexpensive to culture and induce. A plasmid system called PET2NE1 will be used because it has a beta-lactamase gene that allows for ampicillin resistance. Once everything but the protein that expresses DHFR is killed, amplification and induction will allow for the expression of the gene.
The first step is cell culture and induction. The objective within this is to transform bacteria and selection of transformed bacteria. In order to do so, you need the following materials. You need the E. coli, the plasmid, the LB plates, one that has ampicillin and one that doesn't have ampicillin, an LB broth, cell spreader, a shaking incubator that's set at 37 degrees Celsius, warm water bath, and ice bath, and microcentrifuge tubes for this. To start the process, you have to add 5 microliters of plasmid to 50 microliters of E. coli that's in a test tube and let it settle for 5 minutes at room temperature. Once it's settled, you transfer the MC tube that has the E. coli and plasmid to be heat shocked that's set at 42 degrees Celsius for 50 seconds, and then transfer it to an ice bath for 5 minutes. Once after that, of course, you add 1 milliliter of the broth in the MC tube containing the E. coli and plasmid, like I said, and transfer it to a shaking incubator for 5 minutes. Once after the 5 minutes, you can pick a colony within your plate and use a cell spreader to streak 50 microliters of bacteria onto each LB plate. And then you're gonna incubate it overnight at 37 degrees Celsius, and then after overnight, you return to take pictures and determine which LB plate consists of the ampicillin and one without ampicillin. Step to this is the overnight cell culture. Within this, the objective is to amplify the transformed bacteria that were used in the previous step. In order to do so, the materials you need is the plates from the previous step, of course, the broth, and the shaking incubator that's set at 37 degrees Celsius. To start the process, you need to use the LB plates that were already prepared and containing the transformed bacteria and inoculate it in a prepared test tube with 3 milliliters of LB and ampicillin broth. And once you do so, you transfer that test tube to a shaking incubator that's set at 37 degrees Celsius to stay overnight. You then, of course, take a picture of what it looks before you incubate it, and then the aftermath of overnight incubating.
The third step to this experiment is subculture and induction. The objective is growing bacterial culture, inducing expression, and harvesting proteins. The materials needed for this, you need the Langley dye, IPTG, the microcentrifuge system, the tubes from the microcentrifuge, the overnight culture, the LB, and ampicillin. To start the process, you need to add 12 milliliters of the LB and ampicillin to the 1 milliliter of overnight cultured. Once you do so, you then observe and measure the OD600, just like in the graph right next to me showing the absorbance.
Note: before the cells can be induced, the OD600 measurements will measure the absorbance of the culture to make sure the cells are in the optimal growth phase, which is normally in the range of 0.6 or 1. Once induced, the cells will not reproduce quickly, and after centrifugation, the protein can be extracted from the cell pellet and protein purification can commence.
You then also go to transfer to the shaking incubator, and it will be shaking for 1 hour, while you also measure it every 20 minutes. From there, from the 12 milliliters from the first step, you remove 100 microliters and transfer it to a microcentrifuge tube, and then you will spin it for 2 minutes. After doing the centrifuge, you will remove the supernatant within it. Once that, you will add 100 microliters of the lambda dye into the tube, and then you will be resuspending the pellet that's in it. Once that happens, you'll be storing it into a chamber that's set for negative 20 degrees Celsius. What's being stored is the uninduced sample. Lastly, you will then return to the main culture, and then you'll add 25 microliters of IPTG to it. And once you do so, you can return to the shaking incubator. You can return it to the shaking incubator for 4 hours. And that's the end of that step. Now my friend Gamora will take over and continue talking about the next steps to expressing and purifying the protein. So, bye-bye.
So what's next is collecting the cell pellet. You need a screw cap microcentrifuge tube, 1 milliliter Lamley sample buffer, 20 to 200 microliter adjustable volume micropipettes and tips, and a pen and a water bath at 95 degrees Celsius, and a microcentrifuge with variable speed settings above 16,000 Gs. You'll also need a vortexer and a dry ice bath. Take your tubed sample and centrifuge it at 16,000 Gs for 2 minutes. Then discard of your supernatant with a pipette. Take your 100 microliter Lamley sample buffer and add it to the pellet, which will then be resuspended. Then heat the tube at 95 degrees Celsius for 5 minutes. Store your sample at negative 20 degrees Celsius until your analysis. You're going to need to pellet your induced cells. So this is what you do. You take your 2 milliliter microcentrifuge tubes labeled induced cell pellet with your initials, and then your induced cells will then be pelleted through aliquoting 2 milliliters of induced cell culture into each of the 2 labeled 2 milliliter microcentrifuge tubes. And then those 2 milliliter tubes will then be centrifuged at 16,000 Gs for 2 minutes. Then you will discard your supernatant. And then store your cell pellets at negative 20 degrees Celsius.
As the experiment comes to an end, cell growth of the protein can be analyzed via the OD600 results that were calculated earlier on. But for further analysis of the sample, the protein must be extracted from the cell pellets and purified. Overall, recombinant protein expression involves careful choosing for the cell type, plasmid, and recombinant protein to express the desired gene. Once chosen, protein amplification and induction will allow for the expression of the gene desired.
7, 6, 5, 4, 3, 2, 1. Oh, this you crazy mother.